I'm so excited to be here. Thank you, NanoString, for inviting me back. I was here to launch or be a part of the launch of CosMx last year, and now this year I'm going to talk about how we've been incorporating that into our research program. So I'd like to thank everybody that supports my lab, and I'd especially like to thank Lidon, Ashley, and Alyssa for leading some of the projects in collaboration with NanoString. So in the next 20 minutes or so, I'm going to give you a taste about who and what I do. I'm a translational neuroscientist with one goal in mind. I really want to cure Alzheimer's disease. And as I'm talking about our research questions, I think it will become really apparent why we need spatial platforms that accommodate multiple species, multiple different types of tissue processing, and it needs to be high plex. How we're integrating CosMx into our workflow, and where we think that these technologies are going specifically with neuroscience research. So let's hit the ground running here. Alzheimer's disease is a major problem. It's the most common cause of dementia worldwide, and it has become the most feared health condition of growing older in the United States. People fear getting dementia worse than heart disease and cancer. The reason? It's lethal. It kills more people than breast cancer and prostate cancer combined, and 1 in 3 older adults dies with dementia, and there is no cure. You get the diagnosis and you will pass away with it. So while heart disease has decreased between 2000 and 2019, deaths from Alzheimer's disease have increased 145%. In 2016, the National Institute on Aging and Alzheimer's Association got together and made a very important decision, and that was to not study Alzheimer's disease based on dementia or the cognitive impairment, but to define it by its biology, the ATN framework. A stands for amyloid beta. These are proteins that accumulate outside of the cell. T stands for tau. This is a microtubule-associated protein that accumulates inside of the cell as neurofibrillary tangles. And N stands for neurodegeneration. So even if you don't study Alzheimer's disease, if you look at this brain, a healthy brain on the left compared to the Alzheimer's disease brain on the right, there's dramatic brain atrophy, largely due to loss of neurons. And of all risk factors, age is the #1. But 20 years or more before a patient experiences these symptoms, there are changes in the ATN happening in the brain. So while that is incredibly scary, because I am in that age range, I could have some ATN happening, it also offers a window of opportunity. If we understand what's driving this, we can intervene. And that's what my lab does. We're trying to understand what is this black box of aging that can take a young, brilliant mind, that is my maternal grandmother on the left, her high school graduation picture. She graduated class valedictorian, and through the course of your lifespan, turn it into a dysfunctional organ where you don't remember your family. And the picture on the right is my grandmother with me as a high school sophomore, taken shortly before she started losing her memory, and she passed away with this disease. So how do we do this? Well, as a translational neuroscience lab, we take multiple approaches. The first is to understand biology of aging. We take lessons from this discipline where they have identified fundamental cellular and molecular processes that not only co-occur with aging, but if they're perturbed, you accelerate aging. You can accelerate pathologies. And if you intervene upon them, you can delay pathology, you can prevent disease, you can extend healthy lifespan. When we've identified one of these hallmarks that we think is important for Alzheimer's disease, the next step is to determine, is this actually happening in a patient brain? So this cartoon is of a scientist sitting at their computer. The Alzheimer's disease field is big. We generate a lot of data. There's a lot of opportunity to analyze data that's already been generated, but if it's not there, we spend a lot of time looking at postmortem human brain tissue. If we find that this biology of aging aspect is perturbed in Alzheimer's disease, we bring it back to the lab. We identify a mouse model that mimics this. We are able to run interventions in these mice focused on clinically relevant outcomes, focused on behavior, MRIs, and if the intervention works, we move this to clinical trial. So in my lab, we've identified tau as a driver of cellular senescence. There are neurons in the brain with these tangles are senescent in postmortem human tissue. We can clear these cells in transgenic mice, and we're now testing this in a phase 2 trial. But we really don't understand how is tau connected with cellular senescence. We're now ready to move beyond phenomenology and improve these treatments. And our lab is really focused on tau because it is the most common inclusion among all neurodegenerative diseases. Shown here is a laundry list. And this is a protein aggregation problem. Interestingly, if you look at the neurons in the brain that are arguably the most stressed, these neurons with tangles, they're not the ones that die. So shown here is an image of a neuron from postmortem human brain stained for the tangle. This is heavily phosphorylated tau, but these neurons are predicted to survive in the brain for up to 20 years. Yet they're the closest correlate with disease severity, meaning the more tangles, the worse the dementia, the worse the neuron loss. But neuron loss far exceeds the number of tangles by orders of magnitude. So these tangles are in the right place, the right time, seem to be causing problems, but they're not dying. And this is similar to what happens with cellular senescence, which is a hallmark of aging. Cellular senescence is a complex stress response that allows damaged cells to survive. So instead of a damaged cell undergoing cell death or continuing to divide and become cancer, it changes its cell fate. It changes its morphology, it changes its transcriptome, its proteome, and it can't die. The only way this cell is removed from your body is by the immune system. So it actually sends out signals to tell the immune system, come clear me. These are chemoattractants. But if the immune system doesn't hear this, these pro-inflammatory molecules become toxic to healthy neighboring cells or transform cells to become senescent and continually perpetuating this inflammation. So we had a very simple question to ask. Are neurons in the brain with tangles senescent? And you can see this is a spatial question. We need to know what's happening in the cell, what's happening in its environment, and it is a multiplex question. Senescence is a stress response that causes a change in cell fate. So at this point, my lab became really excited about GeoMx. So we now have a project where we are trying to understand when in the adult lifespan senescent cells first appear and if they change. So we are profiling postmortem human brains aged 20 to 100 plus, people passed away aged 20 to 100 plus, with or without dementia, focused on the hippocampus, this brain region that's important in learning and memory. And so this is an image of a GeoMx image of a postmortem human brain. We're focusing on two cell populations within this region, CA1 neurons and entorhinal cortex neurons. And if you zoom in, we're really interested in neurons with or without tangles. The tangle marker is magenta. And not only those neurons, but also their immediate environment. We're measuring 84 proteins in each of these ROIs, as well as the whole transcriptome. And I'd like to point out, in this panel, there are multiple different antibodies for tau and phosphorylated tau. And here's a snapshot of some of those data. So this is a heat map of 324 unique tangles from 11 postmortem brains. Each column is a different tangle, each row is a different protein. And we had a question to ask. What if we stratified this on dementia status? So even though tangles are the closest correlate with dementia, not everybody with tangles actually has dementia. And when we did this, we found evidence that there is less senescence in people that are resilient to this neuropathology. Their neurons had higher synaptic protein expression, and their environments had lower inflammation, lower oxidative and energetic stress. And I'll draw your attention to that purple band. Those are the phosphotau's. We found 2 phosphotau's that were different in the tangles of people that had dementia or did not. But are these cells really senescent? And the reason we ask this question is because in the biology of aging field, there's a lot of publications on senescent cells using single marker genes. And in 2019, the International Cell Senescence Association published this paper and said, if you want to call a cell senescent, it has to have these features. There needs to be cell cycle withdrawal, macromolecular damage, dysregulated metabolism, and the secretory phenotype, which is great, but how do you measure all of that in a cell and a tissue? So I started collaborating with Dr. Habil Zara, a bioinformatician and a graduate student in his lab, and they created eigengenes, which allowed us to look at multiple genes as a weighted sum, and then start asking this question. So we had an eigengene for cell cycle withdrawal, 22 genes, an eigengene for the secretory phenotype, 48 genes, and an eigengene that covered both the dysregulated metabolism and the macromolecular damage with 44 genes. And then we did a lot of bioinformatics, looking at single nucleus RNA sequencing data. Are there senescent cells? What are these cell types? Proving again that they were overlapping, that we had really identified them, weighting these genes, who is the most important. Finally, so we could look at it in tissue, a single marker gene looking at RNA scope, and then the protein with immunofluorescence. We finally published that we think that we really have these senescent cells, and this is all great. But we call this BC, before CosMx. Wouldn't it be great if we could have just started at the end, looking at all of these genes in a tissue? And so now with the birth of CosMx, we're moving AD, or Alzheimer's disease research, into the future. And so for the remainder of my talk, I'm going to be sharing with you all of the rest of the data are going to be from CosMx. We use both mouse and human tissue, and the question that I asked, wouldn't it be great if we could just look at each of these senescence genes in the tissue? Well, last year, we showed data from mouse brains using the 1,000 plex RNA panel, profiling 330,000 cells, 99% past QC. We showed all of these different cell types and the niches, but now we're mapping these transcripts for senescent cells back to the tissue, zooming in, the control on the top, the tauopathy on the bottom. On individual cells, we see that these truly are senescent cells. How are they impacting their neighborhoods? Better yet, we're able to find them in postmortem human tissue. So using the Human Universal Cell Typing panel, this 1,000 plex RNA panel, on postmortem human brain, you can see there are 2 human hippocampi here. And zooming in, looking at all of these different pathways that are covered, many of them have senescence genes. And this is one example of many of analyses that you can do that our lab is really excited about. So we call this the bullseye approach. The red is the target, that is 10 microns within a tangle. So essentially that is the tangle. So anywhere on this section where you see red, that's a tangle. The green is 50 microns around that tangle, and the blue is greater than 50 microns away. So we are able to look at gene expression in relation to these tangles and determine, are senescence genes going up the closer you get to tangles or not? What are other genes doing? And with this technology, we're able to identify for the first time confidently senescent cells in the Alzheimer's disease brain. And the National Geographic issue in January, just this past month, featured living longer and better. So they had scientists that were focused on aging and longevity and featured the work done by our lab in collaboration with NanoString. Li-Dan Wu took this beautiful picture, and Dr. Dirk Keen from the University of Washington provided this brain tissue. And National Geographic calls us the zombie slayers. So senescent cells are sometimes called toxic zombies. We've identified them, and our clinical trial is working to clear them. So now in the next few slides, I'm going to just give you an overview of where we're going with this. So shown here is a healthy human brain on the left, an Alzheimer's disease brain on the right. This is CosMx data. Each green transcript has a corresponding GeoMx protein target. So you can do a serial section. You have subcellular localization of these transcripts. What is the protein doing? We're also really excited about using this for drug discovery. So now that we're able to identify these senescent cells based on the transcripts, we have thousands of other transcripts that we can start to look at and find unique signatures of these senescent cells and develop new drugs. So you just heard about the ultra-high plex. We ran this on postmortem human brain. We were able to detect over 4,500 genes in these tissues. And so now what? What's next? Essentially everything. There are so many questions that we can finally ask and answer. One is comparing the single-cell spatial transcriptome across diseases. So we recently ran this on postmortem human brain from a person that passed away with Alzheimer's disease, comparing it to somebody with ALS and the control. And shown here are the differentially expressed genes, and you can see the patterns are very different. We're able to look at senescent cells across diseases. Alzheimer's disease, ALS, control, you name it. Are the senescent cells in different diseases the same? Are they different? Are they different in different brain regions? None of these questions that we know, but we can now answer. We can now look at subcellular localization. So we've already seen that there are transcripts that are localized within the nucleus, others that are surrounding the cell. Back to what Joe was talking about with these ligand clusters. So shown here is an ALS brain, and the ligand clusters, this bright yellow heat map area, these are ligand clusters in the gray matter. So this is where the cell bodies are. If we compare that to a control, you can see it's dampened. While there are other clusters that are primarily in the white matter that are not existent in the control. So we think that this is going to inform incredibly on what is happening in these different diseases. So gene expression is great. We're so excited about the high plex. But many neurodegenerative diseases are proteinopathies. Well, NanoString is making major advancements there as well. And what I'm going to show you in the next few slides are data that we've generated on mouse brain from the 64 plex. Something that's really exciting to me, because I am a translational scientist that also uses mice, there are human-specific antibodies against proteins that are common to our transgenic models, meaning that we can look at both protein, or at both human and mouse proteins within these models and see how they're interacting. So some of these data are just proof of principle. We're able to see specificity of the human tau antibodies, recognizing human tau only, not cross-reacting with mice. Looking at 138 fields of view, about 89,000 cells, 64 protein targets, all these clusters, what I'm going to point out is tau phosphorylation. So there are 5 different phosphotau targets in this panel, and you can see where they're spatially resolved. We've also identified that there is one phosphotau that we don't see in wild-type mice. It is unique to the tauopathy condition. We can start to look at how these phosphotaus are localized within the cell, how they correlate with other cell types. So shown here is one of my favorite images because it is all tau. It's all of the different tau antibodies along with GFAP, which is an astrocyte marker. We zoom in here, and now we have co-localization of all of these different phosphotaus, and you can see they're not all in the same place. There are specific phosphotaus in specific areas of the cell, which we didn't know before, and we think this will really help inform on the biology of disease. So circling back, we are a translational research lab, really using the biology of aging to motivate therapies to get to the clinic. We're focusing on tau and cellular senescence. And so how is our lab using these NanoString instruments? We use GeoMx as the workhorse for hypothesis testing. When we're after a question, we can run a lot of samples through GeoMx, a lot of different tissues through GeoMx, a lot of different ROIs. And then for validating these hypotheses, we use both GeoMx and CosMx. They're not one and the same. One doesn't replace the other. They're both valuable. CosMx is incredibly useful for hypothesis generation. We get tons of data. We're discovering new things all the time. And we're really excited about the CosMx data because we think we're going to be able to discover new drugs. So just to summarize, GeoMx and CosMx are significantly advancing neuroscience research, and you've heard from everybody on the panel today about all of these things, and I will just repeat them. The data are very high quality. They're repeatable, it's consistent, there's species flexibility, human, mouse. We also use non-human primates in our lab. We've successfully run GeoMx experiments on them as well. They're compatible with all sorts of real-life tissues. We rely on biobanks for our postmortem human tissue, and we are getting incredibly good data from them. We need multiple analytes, gene expression, protein expression. We've already discovered that not all tangles are the same. I didn't emphasize this enough, but we're the first ones to really show that. It was believed for the longest time that a tangle was a tangle was a tangle. They're not all the same. We need tissue-level analysis with subcellular resolution. We're able to see multiple cell types, brain regions, neighborhoods, both mouse and human, spatially resolved gene expression, and spatially resolved protein expression. So incredibly excited about these technologies and where they are pushing neuroscience research. And with that, I'd like to acknowledge everyone that has helped with this science and that will continue to help as we move forward. And a huge thanks to NanoString, Joe and your team for developing the technologies that are going to allow us to cure these diseases that currently are untreatable. Thanks, everyone.
This is how we do it.